Hey guys, welcome to AP Biology, and we're going to be talking about membrane transport, part two. Okay, so again, why do things need to cross the membrane? Just like the cartoon says, to get to the other side. And that's what we're going to be talking about, how things move to the other side. All right, so first of all, we're going to be talking about the plasma membrane, which is a selectively permeable membrane, SPM. So whenever you see SPM, we're talking about a selectively permeable membrane. And permeability depends on four major factors. Factor number one is size. Small molecules pass easier and faster. Number two is polarity. Small nonpolar molecules travel faster. And if you forget what the definition of nonpolar and polar is, I suggest you look that up because you need to know that. Charge. Ions cannot pass through a hydrophobic region of the plasma membrane. They cannot charge through it. It's a function of the membrane structure. And then concentration. The greater the concentration gradient is, the faster molecules will move through the membrane. So if you look at this picture right here, we see small uncharged molecules. Now, water is polar, and water can sneak through the membrane. However, it does it also slowly. And there are actually special transport proteins called aquaporins that help to move water. And it's relatively, I don't want to say really relatively, I guess it is really pretty recent, like the last 10 years maybe, that the importance of these aquaporins has come to light. But glycerol, which is a very lightly polar molecule, it's got slight polarity. Nitrogen gas, nonpolar. CO2, nonpolar. Oxygen, nonpolar. Ethanol, very slightly polar. Some of these three right here can diffuse, although via very slowly, whereas these three can really rapidly move through the plasma membrane. Large polar molecules, they're restricted by size or charge in ions. Ions cannot go through, and strongly polar molecules such as this amino acid and water really cannot move through very well. Okay, so those four things, make sure you know what those four things are. Okay, so membrane transport can be divided into two major categories, passive and active transport. And passive transport, as I say, no energy is required, and it is the movement from areas of a high solute concentration to areas of low solute concentration. Therefore, it's moving down its concentration gradient. Okay, so it's almost like a rock rolling down the hill. It's moving down, no energy is required. Active transport requires energy in the form of ATP. You're going in the opposite direction from a low solute concentration to a high solute concentration. You're moving against the concentration gradient. So you're pushing the rock up the hill, or another analogy, passive transport would be floating down the river, whereas active transport would be swimming up the river against the current. Okay, it requires a strong input of energy. Okay, so diffusion is really straightforward. It's pretty simple, and it happens in non-living and in living systems. And I'll basically take a lump of sugar, you drop it in the glass, and eventually that sugar molecule will diffuse throughout the glass so it's equally distributed. Okay, and it's due to random motion. Another example would be like putting a drop of dye in some water and watching that dye diffuse all throughout that water, or even smelling somebody who walks in the room that has maybe some strong perfume or cologne on. If they walk into a door, the person closest to that door is going to smell it first, and the person furthest away from that door is going to smell it last, as those molecules diffuse from a high concentration, the person wearing the perfume / cologne to people far away. Okay, and it will continue until equilibrium is reached. Once equilibrium is reached, movement still continues. It's just not going in one direction. Okay, it's not going away from the high concentration. It's all over the place, moving everywhere. Okay. Passive transport in relation to cells is going to be diffusion across a membrane. Okay. And again, here we see two pictures. We see a selectively permeable membrane. We see the high concentration on the right on the left in the top picture. And the movement is going to be random, but eventually they will move until they are equally distributed throughout the container with that semi-permeable membrane. Now, the one thing I do want you to notice is that every solute will diffuse down its own concentration gradient. And that is illustrated in the bottom picture, where you see a high concentration of the purple on the right and a high concentration of the green on the left. And over time, they will both diffuse down their concentration gradient until equilibrium is reached for both of these solutes. Okay, lots of questions pop up on that, so make sure you understand that. Okay, facilitated diffusion is basically the same thing, except it uses a transport protein in order to move across the cell membrane. And it uses a transport protein to move things that cannot diffuse through the membrane. So things like ions and large molecules and polar molecules and things of that nature. But there are three major points to facilitated diffusion. Number one, it's specific, meaning that a aquaporin will only transport water molecules, nothing else. A glucose transporter will only transport glucose molecules. A sodium transport only transports sodium, so on and so on and so on. They have specific shapes that allow those specific molecules / ions to move through them and nothing else. It's passive. So facilitated diffusion, once again, does not require energy, and you're going from a high concentration to a low concentration, not the opposite way around. And it saturates. What does that mean? It means that facilitated diffusion can only go as fast as you have the number of proteins. So let's say we have a sodium transport proteins and we have 100 of them. As sodium builds up, it starts to move through those different proteins. Once all of those proteins are active in transporting sodium, the rate of the movement of sodium cannot increase because they are all involved in sodium transport. So that's what it means by saturating. It'll only go as fast as the number of proteins will allow that diffusion to occur. Okay. Again, we also have, again, the idea that it's highly selective. Each transport protein will only transport a certain type of molecule. And once again, it's based on concentration gradients and voltage gradients also, which I'll talk about a little bit later. Facilitated diffusion, once again, here's just a picture of it. And we have a carrier protein. It's a transmembrane protein, and those molecules are going from a high concentration outside the cell down to a low concentration inside the cell. It is diffusion because it's going from high to low. There's no energy required. It's facilitated because a protein is helping that process along. Okay, and again, driven by concentration gradients. Osmosis is simply the diffusion of water. Okay, so water will move from an area of high concentration to an area of low concentration. Now, what is a high concentration of water? That's oftentimes going to be determined by the solute concentration, how much of different things are dissolved in that water. The more things that are dissolved in the water, the less free water molecules there are to diffuse down. Okay, and we're going to do a couple of labs on that this week. So the term for the amount of solute in a aqueous solution is called the osmotic concentration. So a solution that is hypertonic has a very high solute concentration compared to something else. And usually that something else is a cell. Okay, so if you have a cell in a hypertonic solution, the solution outside of that cell has more solute in it than does the cell itself. The opposite, if it's hypotonic, okay, a hypotonic solution has a low solute concentration. So a cell that's in a hypotonic solution has more solute inside the cell and less solute in the solution around it. Hypo means less, hyper means more. Of course, if it's isotonic, equal, then there's an equal concentration of solutes on either side. Now, here's the key point. Water moves from a hypotonic solution to a hypertonic solution. And again, that's referring to the solute concentration, because if there's a lot of solute, there's less free water. If there's less solute, i.e. a hypotonic solution, there's more free water. So water does diffuse down its concentration gradient, but in relation to the solute, it goes from hypo to hyper. Okay? So here's an example of both plant and animal cells. An animal cell in a hypotonic solution means that there's more solute concentration in the cell, so water is going to move from outside where there's less solute, more free water into the cell. That's going to cause the animal cell to burst. That's different than a plant cell, because a plant cell has a cell wall that will not burst. Plant cells like to be in a hypotonic environment. Okay? Animal cells like to be in an isotonic solution, where there's an equal amount of solute both inside and outside of the cell. So our plasma that we have in our blood is basically, and in between all our cells, has the same solute concentration as the cells themselves. Plants don't really like that. They become flaccid, i.e. they start to wilt a little bit in a isotonic solution. In a hypertonic solution, both of these, both plant and animal cells, will lose water content, and the animal cell will shrivel up and most likely die, and the plant cell will do the same. We did this in the lab with the microscopes the other day when we put the salt concentration. We saw the cell membrane peel away from the cell wall of the algae there. So talking about osmosis, here we have a 3% salt solution. We have distilled water. So this is hypertonic in relation to the environment. We put this into the water as a selectively permeable membrane. Water's now going to go rushing in, trying to equilibrate the concentrations on either side of this membrane. Now, it can't quite do it 100%, and we'll get into that a little bit later tomorrow when we talk about water potential. But as you can see, as water moves in, we see the water level going down, the water level here going up. We also see it going from dark green to light green. It's becoming less concentrated there. Think about a salmon. Okay, salmon are fish that are born in fresh water. They grow, they spend most of their life in the ocean, and then they return back to fresh water in order to lay their eggs. And of course, that's where they will die also. That's an incredible physiological change for that fish to do, to go from fresh water to salt water and then back to fresh water. And it's so great of a change that the fish really can only do it once, okay, because they do die the second time they do it once they are adult. They can get by with it when they're young, as the bodies are a little bit more plastic, they're still undergoing development and so on. And that is it for today's lecture, and I hope that helped, and we will see you later.